Hello, everyone. Good afternoon, good evening, good morning to everyone who's joining today in this special session of the Brazilian Symposium in Bioinformatics. Thank you very much to all the organizers of this great symposium, and thank you for inviting us to have this special session. So I want to talk about the RIA Bio, which is the sponsor of this session, and I want to tell you a little bit about what RIA Bio is. So if we can start the presentation. So thank you very much. For those who don't know, RIA Bio was founded in 2020. It was an initiative of several countries in Latin America, in Iberoamerica, with many researchers, students, and also companies. That was funded by CITED to have this project running from 2021 to 2024. Please, next one. Okay, so RIA Bio, as I told you, it's now formed by 172 researchers. It's 35 groups, 65 institutions, and there are also several companies and startups. And at this moment, there are 10 countries that are part of this initiative. RIA Bio means, so in Spanish is Red Iberoamericana de Inteligencia Artificial and Big Biodata, which is International Network of Artificial Intelligence applied to big biodata. This initiative is to be able to share between the countries all the facilities and the expertise of the researchers, and also to get closer to companies with whom we can collaborate. This our leader of RIA Bio is Elizabeth Tapia, who is in Argentina, but we are having a very horizontal group, and we have meetings every more or less every month or every two months, and we want to invite also other countries who will be interested in joining RIA Bio groups or countries, institutions to join this initiative. You are all very welcome. Please, next one. So we already had several meetings, like online meetings, but we finally had our first annual meeting in this 2021, which was held last week, November 18 and 19, and it took place in Salamanca, Spain, and we had a hybrid meeting with some presentations online and some in-person presentations. and we had about 40 presentations, and it was a really fruitful meeting. And this has allowed us to get to know the work of other groups and to start having some collaborations among us. um Next one. So I just want to take the opportunity to invite you all to the next annual meeting of the RIA Bio, which will take place in Querétaro, in Mexico, in November 2022. And this will be also part of the ICCB, the International Society in Computational Biology, together with the International Conference in Bioinformatics organized by Mexican Society in Bioinformatics, and with also SoyBio, which is a um, Ibero-American Society also in computational biology. So if you want to follow us, here we have our handle in Twitter. And so RIA Bio is the sponsor of this session in this moment of artificial intelligence and bioinformatics. And the idea of this session is just to um, showcase what is the importance of artificial intelligence in bioinformatics in this very moment, due to this great amount of data that biological problems generate. We realize that artificial intelligence starts to not starts but has started to play a very important role now in bioinformatics. So in this moment, we will have 5 short presentations by people from different countries. And the idea is that after the presentations, of course, after each presentation, if you have a question, please just write it down, and we will let the speaker know. But it's important that at the end of the presentations we stay here and we make a discussion of how you think or how you see artificial intelligence um, going how this is going to help the field of bioinformatics, or what we should uh, take into account when we use artificial intelligence, and when we do bioinformatics. So please stay with us in this interesting discussion. So thank you very much to everybody who has joined. And if you are here just by chance because you are mistaken, just please stay. It's going to be fun. Um, so we will start with our first presentation that is going to be by Flavio Spetale. Flavio Spetale is an assistant researcher in the bioinformatics group in CIFASIS in Argentina, and he will talk about factor graph annotation of long non-coding RNAs. Thank you very much, Flavio, for accepting to talk about your work with us in this special session.
Thank
Welcome.
you. See, this presentation is okay? Okay. Good afternoon, good evening, or good morning to the audience. My name is Flavio Ezequiel Spetale. I had PhD in computer science, and I work in bioinformatics in groups of CIFASI Rosario, Argentina. Before starting my speech, I would like to thank Maribel Hernandez and the Brazilian Computer Society for the invitation to this event. It's very glad to see for all here. This speech I will taking about the Go annotation, the long non-codings array using factor graphs models. Thirdly, I will have a conceptual framework to the problem of the functional annotation with long non-codings array. Next, will briefly explain what gene ontology is. Then I will cover hierarchical classification method based on orthograph works. And finally, we will discuss further improves and could be incorporate my method. Okay. The first question is, why is the annotation of long non-coding arrays is important? Okay. The study of long non-coding array, more 200 nucleotides, is central to understanding the development and progressing mind complex dishes, for example, Alzheimer, Parkinson, and Brecht. Unlike, unlike the proteins, the functionality of long non-coding RNA is only subtly encoding in their primary secondary. In long non-coding RNA is not here. The functionality is relationship with the secondary structure. Therefore, the secondary similarity method, for example, BLAST, are not directly applicable because this method is not accuracy good for the annotation long non-codings in this case. However, the in silicon methods annotation, for example, inference to interaction network, PPI, is bad because the extensive experimental studies are required by this network. In these other works, this method is very good when you take a lot of samples annotation, but independent the organism. Therefore, in the community scientific search, the need the arise, the develop computational method for the annotation when the starting from the sequence then and the end of the set of functionality. Where, for example, virus, for example, maturity in plants, for example, immunity in humans, and very old functionality do you think in the heart. Here, briefly, namely, Gene Ontology Consortium provide an ontology of terms for describing the properties of functionalities, bien, and their relationships in species independent of the mark. Is in this work, this annotation is not dependent on the organisms. For example, here, molecular function, the father is the catalytic activity, is not dependent the organism, for example, human, Drosophila, Arabidopsis, etc. It's okay. Go is divided to 3 subdomains. Molecular function, here, molecular cellular component, and biological process. This subontology is characterized to 3 subdomains. Cellular component is characterized the cellular compartment, molecular function, the functionality of molecular, and biological process is a set of the molecular function annotation. It's important in this picture, these 2 things. The first, the functionality of the different subdomains, here, can be connected through the links, black, blue, right, and dot lines, is different relationships in annotation. This annotation can be between molecular function and cellular component, for example. Another inner, the subdomains. The second is important to see is another details we observed we descendant on the ontology here, descendant, the color blue and white is indicate the number of the examples. Is in this word, the top levels annotation, higher number of annotation, but we need you specific Go terms, for example, here o acá, proteolysis, is not more annotation. Therefore, is a problem in depending the method of classification for use. Another problem is in biological process, for example, the another quantity of samples in the top is more disbalanced respectively the another Go terms. Here, the factor graph long non-codings annotation for long non-codings arrays. This model here in blue is not our proposal, is divided in two steps. In the first step, here, characterization 2-second plus, and is an encoding-based approach using the fuse, the primary and secondary structure information of long non-codings molecules. And the secondary step is the classification method of hierarchical types. In this word is an classification binary ICBM support vector machines and the support sum product algorithm to check the consistency of the prediction of individuality functionally Go term. The result is a Go term consistency of prediction. We focus in characterization of long non-codings and then the method of hierarchical of long non-codings array. Well, these methods newly, the first step. The first is encoding primary and secondary structure and is transformation to a vector to a fixed length because the method of classification needs a number fixed to inputs. It's okay. For example, here, the secondary structure of long non-codings have 6 substructures: bugle, stems, inner bugles, and etc. In these methods, to incorporate these statistics in the characterization to this plus. And the first step in the method of characterization is the future aggregation. It's no more than the commas to the new alphabet generated to the smile representation is not because the dot bracket and the primary RNA sequences to alphabet the first character R-U-G-I. Yeah, it's okay. Here is the factor graph model. Our proposed the factor graph model allows a graphical modeling of classified systems. That is, collection of the interaction as function. Here, G and F are function, and the circle is nodes, observable and latent nodes. Observable is provided to the support vector machine, and latent node is the Go term. Here, the results in the Go consistent predictions. Okay. The relationships, for example, parents and child, is encoding and into the function. In this case, is logical function. She is the modeling the probability distribution function. This word is the real support vector machine. In this word, es decir, that the noise is represented for the function. Perfect. On this solution, a graphical model is obtained in what is possible to carry out in the inference by the missing passes algorithm, such as sum products. Here, the inference is the belief propagation of some product. Some product is the generalization of belief propagation methods of factor graph. Here, the logical F3 or H4 added in from the mu passages. The passages direction, for example, G4AX4 or the F4AH4. Another example too. We can mention that the messages function move through the factor graph annotation independently of the each and another situation. This approach a map solution because we are in graph with cycles. Finally, the performance the long non-coding array is presented. Here, for different algorithms, models are not models. Presentation. Daño radio, Homo sapiens, mus musculus y scars for farm. The precision recall and F-score is good, and the hierarchical precision, hierarchical recall, and hierarchical F-score suggested this method is would approximated from the long non-coding array sequences to study another functionality. Finally, the promising accuracy graphics here and here, the range of 65 and 80%, we are observed around the 4 datasets, suggested that the factor graph long non-coding classifier can efficiently annotate from raw long non-coding sequences across the completed full gene ontology Go terms. In conclusion, the factor graph effectively provides an interpretable and scalable graphical model for the Go functional annotation. Thank you for your attention. And here show the research group and the photo of my city. Questions here?
Flavio, thank you very much for the nice presentation about these factor graphs, which
Sorry for my English.
I think many people
here.
I think many people don't know. Flavio, thank you very much for the nice presentation about this fact. So are there any questions, or we just better have a discussion at the end? We can ask all types of questions at the end. Anybody has a question? I don't see any at the moment. Okay. So we continue, and then we have a discussion on all the topics then at the end of the session, okay? Okay, so our next speaker is Angélica Alejandra Serrano Rubio, who is a postdoctoral research at the Center of Research and Advanced Studies in Mexico and the Department of Computer Science. and she will talk about biclustering analysis related to tumor progression of hepatocellular carcinoma. Thank you very much, Alejandra, for joining today this special session.
Thank you very much. Uh, Hello, everyone. My name is Alejandra Serrano, and I'm going to talk about the clustering analysis related to tumor progression of hepatocellular carcinoma. I'm going to start talking about bioinformatics analysis of microarrays. And this kind of analysis is generally performed through the analysis of genetic expression matrix. This kind of matrix generally are stored in different formats, such as database or plain text or any kind of database. So there is no generic way. There is no generic way to store them. However, this kind of data need to be analyzed, and generally this analysis is made up by differential gene expression analysis that consists in quantify the gene expression level in order to determine the relationship between the genotype and the phenotype. Um, Generally, this kind of analysis is made up through the direct comparison of expression level with the aim of knowing their function or implication in some disease and determine the psychological alterations and their implication in pathological process. The algorithms that always we use are based on data mining techniques, and this kind of data mining techniques are focused on group analysis. In these cases, we can identify do two kind of techniques and call it clustering and biclustering. The first one are based on um, find groups of genes or conditions that have a related expression level in all the set of conditions or all the set of conditions. And the techniques and the cluster techniques are based on find out sub clusters that have a subset of genes and subset of conditions that have a related or anti-correlated gene expression level. And in this kind of techniques, a pattern can belong to more than one grouping, than one cluster, or not belong to any cluster. Uh, in this case, these kind of techniques are classified as unsupervised learning algorithms. Is to say there is no a priori knowledge of data set to be analyzed, and this is the main characteristic that the solution needs to be validated through multidisciplinary work. And another problem that have this kind of techniques is that these techniques are considered as NP hard problem. Is to say there is no algorithm in polynomial time that obtains the best solution. Um, um, its execution is carried out as follow. We have an input that can be a database and plain text or any kind of file, and a source code. And this source code is analyzed or is executed in an architecture composed by an operating system, a memory, and kind of hard disk or and a processor. And this source code is compilation to obtain a binary code. This binary code is execute many times to in order to obtain a biological congruence. Is to say the solution obtained by the execution of the binary code considering the input is congruent with the sorry, biological reality. So in this case, we can say that more data, we need more time. The solution, in this case, we need to optimize the runtime to obtain results in less time. So the solution is the use of high performance computing. In this case, we need to take advantage of expert knowledge to obtain a good performance. Our solution is based in optimize the source code using mechanism based on high performance computing as CUDA, OpenMP, and take account the architecture where the source code will be executed. And in this case, we use a MPI to in order to share data between the computers or nodes that compose an architecture. Then we will obtain a binary code that we call as star source code, is to say is the code that will be optimized by mechanisms based on CUDA, OpenMP, and MPI. Consider all the things that are used and compose an architecture. Then we propose an hybrid heterogeneous parallel algorithm design in order to improve computational performance. In this case, we need to classify large high-dimensional data sets. So we need to contribute the refinement of the centroid. We will consider a set of computers linked by a communication network. This set of computers are imperfect and prone to failures. So we need to each computer or node solve a problem of clustering and declustering. Each node or computer have own memory, processor, GPU, and each computer have own hard disk, are and each computer can have the same characters of the other computers or different characters. To solve this problem, we propose a solution based on consensus protocol. In this kind of protocol, we say that each node in a distributed architecture have a status. First, all the nodes or computers have a follower status, and if any node receives an order or a petition about other node, this node or this computer oh sorry this node sorry oh sorry this node request request a vote of all the of a request. Each node requests this node requests a vote of the other node to pass another state as candidate. This then the node that is a candidate pass the next status called leader. The leader is the node. The leader is in charge to orchestrate all the operation of the nodes and the others of performing the operation that the leaders indicate. Then, the algorithm starts with the initialization of all the parameters. Then, through the leader node in a distributed architecture, and until a convergence criterion is met, the parameters or the centroids in this case are propagation to other nodes. Where each of the other nodes or concurrents execute a specific algorithm, like K-means or any kind of algorithm of the clustering. And then when a node or a computer finish the execution of a specific algorithm, send all the results to the leader node. When when a convert criterion are met, the algorithm finish. We did a computational time analysis based on the type of operation performed. In this case, we obtain a gain of 1.33x in a homogeneous architecture composed but 4 nodes. And we can see that we need more computational time when we are going when we did operation related on execution elemental operations. Then this kind of operation are parallelized, consider CUDA and OpenMP. This algorithm was applied to bioinformatic analysis based on the analysis of hepatocellular carcinomas. In this case, our case study was a data set related on the temporal development of preneoplastic nodules and hepatocellular carcinomas. The data set contain was described as a high dimensional data set, and in this case we have a set of experimental conditions determined by the time and in each state of development of the disease, as 7 days, 11 days, 16 days, 1 month, 5 months, 9 months, and so on. And then we select 9 months to analyze, because we have the information related on nodule, not nodule, tumor, and not tumor. Then we have the research question determined by try to understand the hepatocyte biological model. In this case, we want to know the functional classification of genes involved in the tumor development, based on the idea that we have the basal condition and there is something we didn't know what, but this kind of thing that we didn't know, we didn't meet, generates the presence of nodule or the absence of nodule. And if we have a nodule, this nodule is this nodule generate a tumor. In this analysis, we did a pre-processing of the data, and we obtained the 94% of genes that were included in the experiment. So the genes or patterns were removing because this kind of genes were considered as outliers. And in this case, we use a technique called housekeeping. Then we did a statistical analysis, statistical data analysis. So in this case, we can see basic gene silencing based on the baseline condition. In this case, we can say that the gene expression level decrease in the first quartile, and there is more dispersion in the third quartile. So we can see a gene inactivation system for the production of structural proteins. And then we apply the clustering and biclustering algorithms. These algorithms were parallelized. Consider the algorithm that I present in the first part, and we analyze the genes involved in the basal condition, in the nodal condition, and the tumor condition. The 10% that are present in the three condition, we didn't take into account because these kind of genes are present in all the moment. So we analyze the 13 and 18% of genes involved in the presence of nodule, and the gene present in the nodule condition are present in the tumor condition. So we did an ontologic analysis, and we obtained that we obtained that there are a lot genes involved in metabolic function, and in this case we can say that the cell needs a lot of nutrients, and then we need the cell needs to have a metabolic function to generate more nutrients. And in this case, if the cell needs nutrients, so the cell need more energy. This conclusion was obtained by the biological part. So our work only was determined by the analysis, performance analysis, and the in silico analysis. And we obtained a maximum speed up of 3.96x using only OpenMP, and 56.98x using only CUDA. In this way, we increase the traditional optimization guide by a computer expert. We can say that in this work we have focused on performing generic parallelization based only on the parallelization of elemental operation, and without taking account the complexity of this kind of algorithms, and there is not yet a general algorithm that solve the problem of clustering or declustering in biological data. So if we are going to concentrate in a parallelized one kind of algorithm, this solution not be a general solution. So we need to work in parallel the elementary operations to improve in order to improve the uh, performance the algorithms general in a general way. So this is all from me. I don't know if you have some questions, and thank you. So
Thank you very much, Alejandra. This is very interesting. This is something that we have to think of when we do artificial intelligence. We always think like, okay, let's improve it. Let's do artificial intelligence, but we don't take into account all the data input outputs and computer processing and everything that we need. So this gives us an insight of what we have also to take into account, right?
yes, yes, yes.
Yeah. Okay.
Uh,
Thank you. So I don't see questions at the moment, or maybe I don't see them. Do you see any? But I think we will have general discussion about this and the other topics at the end of the presentations. Okay. Thank you very
okay.
much. Okay. So our next speaker is Mirele Carolina Sousa Ferreira Costa, and she's from the graduate program at the University of Brasilia, and she will tell us about investigating the performance of machine learning methods for predicting non-coding RNAs using in silico construction of artificial data. Thank you very much, Mirele.
Thank you. Good afternoon. See my presentation? Okay. My name is Mirelle. I will present about my master research and joint work with my professor Valder, Professor João Victor, my advisor Maria Emilia, and Professor Peter. I will follow this content, introduction. So here we have some examples of no-coding RNAs, and in particular, the small nucleolar RNAs is NORDA. Study this work. Some methods for NCRNAs and prediction use homology-based approaches such as BLASH and Inferno, and machine learning-based approaches such as Snow Report, 2.0, CVM Portrait, and Mustard. The Mustard use artificial neural networks. So our motivation is identifying NCRNAs is a very important task. However, when we refer to the inference of NCRNAs considering large evolutionary distance, which involves research in homology. Machine learning methods did not achieve the expected success. for a systematic investigation of the performance of machine learning methods for homology inference in NCRNAs. We need large, controlled, and known sets of data with evolutionary distance. This task is not simple, as a large volume of reliable NCRNAs that can be used is not now. Oh, the problem. Machine learning methods did not achieve the expected success for homology inference task. For example, to predict classes of NCRNAs considering large evolutionary distance. Objective is creating sets of artificial NCRNAs which cover different evolutionary distance and evaluate machine learning methods in search of NCRNAs homology using the artificial data. And the SNRNAs are of the class of small NCRNAs, and they classified into main types, CD box and HAC box. Their structures are represented in figures and A and B. Machine learning. So, for performance evaluation, we implemented in Jupyter Notebook, TOE, and machine learning models, support vector machine, artificial neural network, and random forest. Data source. I use marine species, Siona intestinalis, is an attractive species for studies on evolution because its development is fast and has a compact genome. So, data processing in order to obtain sequences of SNRNAs within the intro without correlation between each pair of them. Therefore, we retrieve intronic sequence with SNRNAs that are not correlated. Method description or method is divided in two parts: creating artificial SNRNAs and evaluating the machine learning methods. In creating the artificial SNRNAs, we select a CD box and HAC box sequence and generate 4 balanced trees using 4 types of point mutations: substitution, insertion, deletion, and substitution with codons. From there, we built our database of according to the percentage of mutations of the 3 sequences. We extract features from each class and evaluate machine learning methods. Okay, and results and discussions. The build for mutation trees for CD box and HAC box SNRNAs, the trees with 100 brands is children. And database with 5,000 positive sequences and 5,000 negative sequences. We chose the substitution to discuss results in more detail. Okay, BLAST results. We tested the BLAST N tool with the substitution tree sequences. The first experiment with SNRNAs inside the intro we obtained 100% of matches, but these matches are spurious hits in other parts of the intro. In second experiment is known as outside the intro. You obtained matches only in the database with 10% mutations for CD box and for HAC box in 10 and 20%. But where there were no finds with more than 70% of the matches sequence. We obtained similar results with BLASTN regardless of the mutations model. For sets with more mutations, no matches. So, machine learning results for CD box. We achieve good results with accuracy above 99%. The classifiers showed that the more mutations, the performance decreases. These are the Hockey curve of the classifiers in each databases. ANN and SVM obtained excellent Hockey curves with 10% mutation. With 50% the performance was decreased. So we tested machine learning classifiers with a reduced number of features to investigate cases where the biological features are not known. In this experiment, you can see that the features set is quite important for CD box prediction. If relevant biological characteristics are not known, the performance of the classifier is decreasing. These are the Hockey curve is well below the three classifiers. For HACA, the results were decreasing, but accuracy was below 70%. These are the Hockey curves. We also tested with the reduced number of features, but didn't get a big chance in the results. These are the Hockey curves well below. Okay, conclusions. Machine learning methods can be competitive for homology inference tasks if sufficiently large sets of independent sequences can be constructed. Careful construction of artificial data is a processing approach. The comparison with a reduced number of features showed how important non-biological characteristics are important for machine learning classifiers to achieve good performance. Next steps, the perspectives compare the performance of machine learning methods with more sophisticated homology research methods such as covariance models, thermodynamic models, and applying the artificial dataset construction methods to other NCRNA families and evaluate the performance of machine learning methods. And testing different configuration for artificial neural network and compare with state-of-the-art works. Thank you very much.
Thank you very much, Muriel, for the nice presentation. This is also a very different approach of how machine learning can be very helpful for like long-lasting problems in bioinformatics, such as homology detection, right? So thank you very much. We have currently no questions, and we continue with our next speaker. So now we will listen to Lucas Maciel Vieira, who's also a graduate student at the University of Brasilia, who will tell us about machine learning to predict non-coding RNAs. Thank you very much, Lucas, for
Hi
joining today.
everyone, it's a pleasure to talk a little bit about machine learning bioinformatics here. Let me know if you guys can hear me well and see my screen. Okay, so basically today I'm going to have like more than a kind of a small talk, talking about machine learning to predict non-coding RNAs. The main goal here is not to talk about a specific project, but like to build a timeline of projects in which I used machine learning in bioinformatics, and show like the main outline of how we build these pipelines and its results. Okay. So firstly, I will start a little bit like why we should use machine learning. Okay, some of the previous presenters already talked a little bit about machine learning for what is used, and basically it's used to optimize a performance criterion by using example data or parse experience. So basically, if you have a database or data, we try to build generalizations and prediction models to get information from this data. Okay, and why it's good to use this in bioinformatics? Because right now we have several databases, like most of them are open source, presenting biological molecular data. Mainly on this case here, I'm talking about no-code RNAs. We have a lot of databases with no-code RNAs and its disease-related information, right? And several biological studies these past few years have shown that using machine learning is a good choice to predict specific molecular classes or its interactions with diseases. Now I'm going to present here like the works in which I started using machine learning. Basically, I focused there on predicting link RNAs. Previous, someone already talked a little bit of long non-coding RNAs that are very studied molecularly, kind of molecularly nowadays because of its relationship with diseases and etc. And link RNAs are a specific kind of long non-coding RNAs, right? On this specific study that I started in 2015, I built a pipeline using support vector machine, that's a supervised machine learning model, to predict link RNAs in humans and mouse. Okay, at the time we just had like one method that used to do that for link RNAs specifically, that was icRNAs, and basically we try to replicate its pipeline, making a few changes to see if we got better results and how good the results were. And here we can start like seeing the main outline of a machine learning pipeline that I used to build, where I have basically three steps, where we have to build a database with reliable data, remove the outliers. We have to select some features that is going to characterize the data points we want to classify, and afterwards we build the prediction model itself. In this case, we used data from Havana ensemble and got longer link RNAs and protein coding transcripts from humans and mice. We like cleared a little bit the data, and then we started using some features that are supposed to characterize link RNAs against protein coding transcripts. And at the time, we used these three features like conservations, the of length and of proportion, and some G-entry nucleotide repetitions that at the time the studies showed that were like characterizes link RNAs. Okay, so with these three feature sets, we make a study. We made a study with SVN where we tried each feature by itself and build a model with it, and try the combination of them and all of them to see how good they were in predicting link RNAs. So here we could see that the off characteristics was a little bit, it was the most important feature to predict that. But when we combine all the features, we got good results, right? Like near 90%. And it was the first results I had at the time. Okay. And then I kept working on this, and I got out from human and mouse and tried to build a classifier for link-RNAs plants that we didn't have at the time. And we had a specific user cases for sugarcane and maize. So it was really nice to do that. And again, my pipeline followed the same outline, creating the database, getting the features and the results. At this time, I changed a little bit the features because the conservation, it was a kind of a difficult feature to get and only had for mice and humans and not for all organisms. And as the previous work showed, it's not that important when mixing with the other features. So we just used our off length and proportion and some GN3 nucleotides that we got by a tool from our partner from the lab. And with this, it was a nice pipeline where we got an accuracy around 82% for sugarcane and 97% for maize. It was good because we got a really big list of transcripts that would be predicted from our collaborators from University Federal of Rio de Janeiro. And from all the transcripts, the one that we predicted, we also, the ones we predicted, we did a differential expression study for some annotations of that had red strip diseases. And the one that was most differentially expressed was tested in laboratory and was confirmed to be a link RNA and related with the MIR408 that was already known to be related with the red strip diseases. So now it was good because we got the confirmation in lab that our method kind of worked, at least was a good shot. And then I kept going. Until this point, I used a support vector machine. And then I said, oh, maybe I can try a new method. At the time, before deep learning got very, very, very popular, most of the algorithms were using ensemble. That is a machine learning method that tries to combine other methods to have better predictions. And that's what we did. We just follow the same steps, just change a little bit the GN3 nucleotide frequencies and the databases. And by using ensemble, we tried a method of consensus in majority using C3, KNN, SVM, and random forests. And we got like nice results, saying in which the ensemble methods performed better than the individual methods. And it was nice because we also did a feature extraction study to see which feature were more, let's say, reliable or more important to predict, to make these predictions. And here again, we have the off length and of proportion and some combinations of the GN3 nucleotides. Okay, and until this point, I worked with classifying link RNAs, and afterwards, I started to study a little bit more of link RNAs, and I stepped on trying to predict some biomarkers in cancer, right? And these biomarkers in cancers, basically I was studying long RNAs, mRNAs, and microRNAs. And here, although I have an outline pipeline to predict the classes I want or the biomarkers, I got way more focused on doing the biological analysis part because the other projects were kind of questioned because, oh, these GN3 nucleotides, they really have any biological meaning or is just a computational artifact? And I started focusing a little bit more on the biological step where I studied better the features, I built better the database. And here my pipeline was a little bit different, where I got the molecules, constructed computational and networks, checked the function, the biological function of the molecules, the survival curve of the patients related with the molecules. And I did really this just to check if these really have biological meaning at these biological features. And up to now, I just did this biological part, but I'm currently working of combining these biological features that I found with some clinical features and trying to predict with machine learning which of these features are really related to the colorectal cancer. That's my study case. And basically, I'll try to predict the patient cancer stage or its survival curve. Okay, and here's just a picture of the CERNA, the computational endogenous RNAs that I got on this part here, divided by the types of colorectal cancer, and it's a nice picture. And like basically these are the projects that I'm using machine learning to predict no-code RNAs in cancer. Now I'm more focused on getting its relationship with diseases, mainly cancer. And just these are what I got at the end, my biological biomarkers that I use as features on my next step. So I think it's this focus on the biological part to get the features is way more easy, so the biologist that's going to use my work to understand, and I hope it gets good results when I end the machine learning part. Okay, and finally, this is some published work that's related with the works I presented. And up to the discussion, mainly I'm just going to show some points that always comes up when I'm building these models. Firstly, do we have enough data to build these models? Or like the amount of data is so low that most of the models we are going to build are going to be overfitted or not generalized. This is one point. The other is the feature selection, like long RNAs and link RNAs, they were not well known, and even now we don't know like features that really characterizes them. So when we create features using like computational methods that like the frequency of G and tree nucleotides, are they really meaningful or just like artifacts or noise? And that's the next point also, because like maybe they don't have biological meaning, so it's difficult to upset them to predict what we want. And finally, the results validation. Sometimes you have the data on the public databases, you make the model, the prediction is good, but mainly it's nice to know in the lab at least once or twice if it's really working. So this is what I had to discuss, and just an acknowledging of who's supporting my work. And if you guys have any questions, I'm up to answer them.
Thank you very much, Lucas, for the nice presentation. You brought up several important questions that we have to address when we do machine learning or deep learning or in general artificial intelligence. I'm sure we will have an interesting discussion about this when after the talks. So our last speaker um, is João Victor de Araujo Oliveira. He's also a graduate student at the University of Brasilia, and he will talk about Snow Report 2.0, new features, and a refined support vector machine to improve snow RNA identification. Thank you, João Victor, for being today here in this special session.
João, you are muted. Um,
Meeting me?
yeah, now I think that you can hear you. Thank you.
All right. I'm sorry for that. So today I'm going to talk about Snow Report 2.0. It's a tool published in 2016. Uh, uh, and basically, I'm going to start with a background, then present Snow Report 2.0, and finally open a discussion. First of all, small nuclear RNA or SNO RNAs are small no-coding RNAs classified based on their characteristic sequence elements called boxes. We have two here. We have the CD box and 8ACA box SNO RNA. They are classified based his boxes that are motifs that occur in a specific place. Here we can see the canonical secondary structure representation of each of them. They normally tends to fold in this spatial structure, and since it not conserved in primary sequence, it's a very challenging problem to we can use actually machine learning methods. Machine learning methods, I really like this definition. It's a multidisciplinary field, such as math, statistics, computer science, and many others. and to build computer programs that automatically improves by experience. So we can use a variety of algorithms to simulate human learning by exploring patterns in the data, transform them information. This can be viewed on, for example, support vector machine algorithms that we try to build a hyperplane with a maximum margin of separation with two, for example, classes. We can, for example, classify snow RNAs and non-snow RNAs using this technique. So, Snow Report 1.0, published in 2018 by Jana Hartel, it's a tool that identifies two main classes of snow RNA in single sequences, using a combination of secondary structure prediction using Vienna RNA package and machine learning using SVM. The good thing in this tool that does not use information of putative target sites, and it was training only with mammalian snow RNA sequences. The additional part here is that it did not use multiple alignments for identify snow RNA in genome. And in 2016, we published Snow Report 2.0 that improves and extends the original method by extracting new features for both CD and HACA box from, for example, the secondary structure, from minimum free energy features, and others. We're developing a more sophisticated technique in SVM training phase using recent data not only from mammalians but from vertebrate organisms, and we have a careful choice of the SVM parameters C and gamma. And we actually use updated versions of tools and databases used for construction of the original version of Snow Report. It was already published by MC Bioinformatics, if one would like to read. So this method, Snow Report 2.0, starts with the dataset building, where we have plenty of vertebrate snow RNAs, and we use a kind of technique to create two datasets based on the clusterization of the sequences in order to create a way to split these two datasets based on sequence similarity, in order to use, for example, one data set for training and one data set for test, and do several times this in order to check our performance. So with these sequences, we can extract features using this workflow. Basically, we try to find candidates looking for first for the boxes. For example, in HACA box is no RNA candidates. I try to find these 8 boxes using specific position weight matrices, PWMs, and I'm trying to fold the secondary structure between before each box and after after each box until ACA box. First of all, before each box, if you look from the figure, the secondary structure, we don't know what is the start of the sequences in secondary structure. So we can use RNA-L fold in order to estimate the start position. Then we can make some steps to check if the structure folded in RNA fold is suitable, is similar to the ones that we know in HACA or CD box. Since we have a correct structure, for example, in HCA box, a double hairpin loop structure, we can actually extract the features. The same occurs on CD box when we try to find candidates C and D, and in the middle of them, we can check, for example, some structures like intern filter that it appears on the close by the C and D boxes and close to the stand terminal of CD box. If it presents some intrinsic features that is already known on the literature, on the many other research previously, we can fold this secondary structure using RNA fold, and we check if we have a similar secondary structure of CDBox. If we have that, we can extract the features. This is a bit restrict. We restrict Snow report to find very canonical sequences. That's a good point, since we have many known expertise on Snow RNA. They have to present some features to be a Snow RNA, but actually we have a problem. We can find Snow RNAs very different, very non-canonical, like we're going to see on the validation phase. So we start with feature extraction. We extract many features. In blue is the new ones in comparison of the first version. Here we have, for example, some information about secondary structure, GC content, and many others. Actually, in CDBox, we have plenty of new ones here, but the blue ones just vanished, okay? So, extracting the features, we can use actually a Snow report. Snow report works like a feature extractor. Then we can use these feature vectors to create a model that can classify a single sequence in Snow RNA or not. So we use SVM scale on the feature vector. We use the grid search technique to find good C and gamma metaparameters, and you use this to train our model. Our model is trained using SVM train, and then we can have a model that can be used to predict Snow RNAs. And actually, in this workflow, we actually add some performance measures that it not appears in libSVM. We used 3 types of measures. Fixed threshold, where we have, for example, probability to be a Snow RNA or not, like BSVM. And this we can measure by accuracy and F-score. We have dynamic threshold, where each positive sample will be used as a threshold, and we compare this threshold with all the others samples. And then we can estimate average precision and area under the curve. And finally, to check the errors from our model, the discrepancy between our model with the reality, we use residual sum of squares. I'm not showing all the results since it's too big, but from these 2 models we created, from CDBox and ATAC box, we have good results compared to Snow report using the same data. We have the F-score more 14.92% on CDBox, and F-score better 10.9% better than Snow report 1.0. All performance measures is greater than 90, which is good, but actually from non-canonical Snow RNAs, the accuracy is not so good, but it's very nice. For example, here we have in the validation phase some papers that provide some sequences already known of Snow RNAs. For example, in human, we find almost everyone. But in platypus, for example, which is a bit different, I'm computer science, but the platypus is a bit different from human and chicken, for example. And it not finds so many. The same occurs, for example, on the invertebrates. Since our data was used from vertebrate Snow RNAs. For example, on HCA, we have good results, but in CDBox, we didn't achieve good results here. And Leishmania, we will not have good canonical Snow RNAs. They are a bit different, this kind of species. So even in Snow report 1.0 and Snow report 2.0, it finded anything. It didn't find anything here. That's I think it's a good discussion about this. Snow Report was a refinement of a previous version of Snow Report. It's available. I put this GitHub here to download for you to download, but you can just put on Google Snow Report 2 and you find it. Uh, we have see that we experiment with very different organisms have shown good performance, even invertebrates. In the case of ATACA for city box, I think the good way is to retrain, or for example use another kind of features. That's one idea that I have, that to create specific data sets to identify better features for different kind of organisms. For example, we can use Snow Report to train and create a model for Leishmania HACA boxes, for example. We have to use different approaches to fold the sequence and different machine learning methods. For example, we can use math-based feature extraction like complex networks, or using some, for example, Fourier transforming to create a model that will be, I think will be more suitable to identify non-canonical secondary structures of snow RNAs. I think it's a good idea. Actually, we already use some complex networks on this and presented a better result. But Leishman is still a question mark problem. Uh, Another thing would be very nice with this project is to create a parallel version of Snow Report, since they can be used to screening a genome in order to find many snow RNAs. So this is my work. We have some papers very interesting in this field, and thank you all for this opportunity.
Thank you very much, Vitor, for this interesting update in Snow Report, and for putting us up to date as well on what are the things the challenges that we still have in the in prediction. So if we want, maybe now we can open all the people, the cameras and microphones, and we can talk about some, some of the challenges that we have in artificial intelligence. Raquel might also want to take part of this because she already suggested a couple of topics, right? So it would be nice to have you, Raquel. Thank you. Yeah, so Raquel was already suggesting that for the group to discuss about the challenges concerning actually the explainability and opacity of the models. We have heard, I remember since I was a bachelor student, we discuss about this very often, right? That we don't know, like all these artificial intelligence methods look like a black box, no? Or seem to work like a black box because we don't know what's happening there. And so nowadays there is this important discussion about the explainability of the models. So what do you think about this? Whomever wants to participate.
Hi, Maribel. I think that's a very interesting question about how to explain the machine learning methods, for example, for biologists and for ourselves from computer science. I think, for example, my Snow report uses previous biological knowledge to find snow RNAs. But however, we see many other methods, recent methods that use, for example, mathematical based feature extraction methods. that is a bit difficult to understand what is this meaning, what the machine learning method use it to say this is a snow RNA, for example. This is a very good question. I think it's a challenge we have now.
Yeah, yeah, this is true, and also as Raquel mentioned, especially now for deep learning, no, like in deep neural networks. So what do you say about this, Raquel?
Yeah, I think that one point that is sensible is the healthcare system that is also trying to use machine learning techniques to support in the diagnosis and things like that. So I think that it's an open problem still, isn't it? We like João Vitor mentioned that this tasks involving feature extraction, feature selection, maybe they can help. And another point that I add here is maybe that visualization techniques can also be useful for a more deep understanding of the algorithms and the model construction. I think that is something that we have to deal with in the next years if we want to use artificial intelligence models for healthcare in biological sciences, life sciences in general.
Yeah, that's true. Well, it's also not easy to visualize this, no? How so, for example, how a neural network works. So even though there are many models out there and we can see them like these recurrent neural networks or neural networks, even though we are computer scientists, we don't we not all of us are experts in the functions, optimization functions that the deep networks use in every layer, for example, right? And it's not that easy also to explain and to understand. Like you have to really work in this field, or like many theoretical computer scientists are the ones that really understand how all works. But it's true that it's hard. I don't know. So if someone else, if someone has worked with deep neural networks, what you think about this?
Yeah, like uh, when we are talking about this subject, about how important it's to understand and to explain what we are using, like I had a lot of these problems when predicting long RNAs and microRNAs, because like we already don't know many biological features that characterizes it. So when you get the machine to create new features, sometimes the combination of features that we are understanding is really difficult to pass to the biologist or even to the doctor that this is really a real feature. And nowadays on the computer science, like right now, we just are using basically deep learning, where we have a lot of data. We just create like features and we accept the results because most of the tools that uses machine learning with deep learning work is now. But we have two main problems when applying this to bioinformatics, as I see. First, we don't have enough data for most of our pipelines. Like I think it was Daniel et al. That say that we need at least five data points for each group to start to perform well with deep learning, and mostly we don't have that. And the second is like how we are going to make like health apps, like Raquel said, or to explain this to the biologists or to the doctors. So sometimes I try to use like feature reduction instead of a dimensionality reduction, so I can keep the features as they understand, even if my model doesn't perform that good. So
Thank you, Lucas. Yeah, this is true. And then there is also one more thing that actually Alejandra brought up brought up in her presentation, right? That you also, so sometimes you don't have enough data. Sometimes you don't even have enough computation process, like power to do it, right? Or storage, even if you have the data, right? So what do you say about this, Alejandra?
So in this case, I think that not all of can be expert in all in all the area. So, so as you can say, there are a lot of problems in large databases and the dimensionality and other kind of problems. So we must promote the multidisciplinary work, I think, and I I am confident about that, and we must involve in other areas as computer science expert. I think that the biologist is an expert in biology, and we need to help 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 them to improve their analysis. Or I don't know. And as we say, there is no general solution to solve any problem. Yeah, the biological problem. So all the problems are really different, and we must work in a multidisciplinary form. So,
Thank you. There is also a question here or a comment from Marcelo Reis from Butantan. He says, for some problems, one workaround for the interpretability problem might be the usage of hybrid models in which one part is machine learning and the other one is a mechanistic description. For example, OD, like ordinary differential equation system that describe the kinetics of chemical reactions. This is true. Actually, I think what Flavio was presenting, it's also kind of a hybrid, right, Flavio?
Yes, the the hybrid models is not good. Is dependent of the organism because not all the organisms, for example, ODE era. I don't know, remember the system in human or and the Drosophila, several fish, a lot of tools with characterization, the RNA and protein, gene products, but when you lost the no organisms models, you need the created and the generated features about the sequences or the another tools prediction, for example, 3D tools. Nowadays, the accuracy of the models in 3D prediction are very poor, but the information is very good. However, the experimental evidence is not increased because the huge volume in the machine learning. Machine learning tools increase and the experimental is the linear progress because the cost and the time consumed to the experimental. In my case, the evidence is very important because the confiability or reliability, the precision is point crucial in the factor graph, for example. In electronic evidence, a lot of nowadays because Blast to Go and others ensemble methods, you annotated new functions, for example, and it's not true this function is real. It's a problem nowadays in the machine learning method.
Yeah, that's true. Anyone else wants to comment on these hybrid models? I think it's very interesting.
Can I
Yeah, Maya,
say
please.
something? Yeah, another hybrid model is the one from João Vitor that he used a pipeline where he used also a machine learning technique there, right? And so I think it's very common to build pipelines that have parts that use machine learning techniques and other parts no. So I think that Marcelo is right, Marcelo Reis. Uh, I would like to introduce some other things here. Although deep learning and machine learning methods and AI in general all of these are being very used, very broadly used. The use of these methods, I think they are very problematic, in fact. They are powerful and they are very easy to use, but as Maribel said, they are black boxes. And so when you use, you have not in my opinion, this is not only with deep learning, but the methods are used in a not very clear way. And so people use methods that are not designed to be used in a particular context. And so I think this has to be addressed. And this is the work of Mirelli, for example, the features and variables that you use in these machine learning methods, the methods are very sensitive to them. So when Mirelli tested this, there are two main characteristics. One is this. The idea when you use more biological features, the methods act and the outcomes, the results are better. And when you don't know, the results are not so good. And so, and another thing is in the methods that she tested, and for the particular non-coding RNA that she used in the test where there is no RNAs, as far as the evolutionary distance, the poorest the results. So there are a lot of biological characteristics that have to be addressed, and usually we are interested in finding out and discovering new results. And I think Lucas also mentioned some of these points. Let me see. To validate the results, a careful choice of input data. And Flavio just said, the data many times are not reliable. And so all of this has to be addressed. And of course, you have to be sure as much as you can that you are really modeling the biological characteristics, in fact, and you are using the right method, and you have to be sure that it was designed to do the things that you think they do. So these are, I think, you have to be very careful to use these methods, although they are very easy to use. This is the way I think.
Yeah, this is very true. So Isabel says she agrees with you as well. I think we all agree with you, and also don't forget that so that they are at the end machines, right? So they are machines that we build, and they learn from whatever we give. So if we give like lots of erroneous data or like messy data, they will learn from this, and then the output will be whatever they learn from this data, right? So nowadays there is the big this big trend, and I think it's very important about the coded bias. I don't know if you have actually watched the movie seen the movie Coded Bias, where there is a bias on all the algorithms that identify, for example, faces, right? Like they identify, they are more accurate when they identify male faces than female faces, or they identify better white people than black people or brown people. And this is because the database is full with mainly male white faces, right? So if we feed our database, and this is the same case with bioinformatics, with biological data, right? So, for example, if one of my colleagues had this problem, but that when he tried to identify certain types of proteins, the method is really good at identifying proteins that have these helix, right? Like, but it's very bad at identifying other proteins. And then toxins are always identified as only as this type of the proteins that have this structure. But it's because the database is full of them, because the other structures haven't been discovered. That doesn't mean that maybe all toxins are of this structure, right? So we have to be very careful, because also it's important to know that we don't have, so one thing is that bias in the data, but it's possible that your data is, so that it's not biased, so your data is bad, it's like not biased, that you can have 50-50, you know, but still there is some bias because of other external reasons, right? So one thing is imbalanced data, that this is when you have more of this data than from this, and that's why then your outputs or your predictions are going to look more like one, or they are going to be better at identifying one type. And this is when we have the imbalanced data, but we can have balanced data and still have some bias. So this is very important to also distinguish, and especially also in bioinformatics, because we are using databases all the time, right? So I don't know if you have actually met, or you have actually meet with these problems during the application of artificial intelligence methods in bioinformatics.
Can I talk? Sorry.
Yeah, sure, Ale.
Yeah, sure.
In data mining, especially in my area, we have some algorithms that allow to capture the main characteristics that help us to select the features that have more weight in a dataset, as a principal component analysis, or I don't know. This kind of algorithms help us to reduce the dimensionality of the data and reduce this bias, uh, or I don't know. And also we apply a descriptive statistics to help us to understand in a best way the data set that we are analyzing. So I say there is no general solution, and this kind of algorithms help us to understand and improve this kind of analysis, especially in biological data, because not all the problems are the same thing.
Right. That's also true. Yeah.
Maribel, there is an observation here from Isabel.
Yeah. So she says, I'm a bit worried about some presentations on what exactly was considered machine learning, artificial intelligence, etc. I would like to collaborate a little bit. I'm completing my degree in data science. Yeah, this is actually very interesting, because 20 years ago, everything was artificial intelligence, right? Like everything was put in that box, as far as I remember, like all data mining, expert systems, these neural networks, machine learning, everything was put in if you remember, in artificial intelligence. But as each of one of these areas was becoming stronger and stronger, right? Like data mining at some point, then it kind of become a field by itself, right? Like it is now data science. or machine learning by itself. And now that it's separating kind of deep learning, creating a new area by itself. But I think like as I learned it, is these all are like artificial intelligence methods. But I would like to hear the younger people what you think it's artificial intelligence, whether there is a difference between artificial intelligence and machine learning, for example, as Isabel is asking. Now, Mirela, or you haven't spoken, maybe you wanna give your opinion.
Uh, it's a challenge to get features that are relevant to machine learning models and in general, and databases that are large and enough to train and test the models. In For Isabel, and using different techniques and for machine learning supervisioned in my work uh, is difficult in database and because the size and is important in science data. In general, I believe that a set of evaluation different metrics is essential and to be um, able to analyze machine learning model in general.
Thank you, Mirel. Mia, do you want to say something about this? What Isabel is asking?
No, I think this is a very, very broad discussion. And so I think that Isabel is a bit worried because, yes, I think that if you make many of these meetings, the reflections and the talks are in many different ways. So I think that what is important of this meeting is to say that these methods, although they are really ready and you can use them very easily, you have to be very, very careful when you take them, and you are the researcher has to be worried about data and everything. And so these projects, they have to be very, very careful planned. You are not supposed to this is a good database, and you don't investigate the quality of this data, and I want to use this method, but you don't know exactly for what this method was designed to do. And these parameters are another characteristic of these methods that are very important to choose. I remember that in all the works of Lucas and João Vitor and the other students that developed projects like this, they the parameters, when you changed them, results were sometimes very affected and very different. And so I don't know. These debates, I think the message, if we can live with one, is that the projects have to be very, very carefully designed to really guarantee that the results are reliable. This is I think, the very good advice.
The important message.
I would like to add
So,
something, if it's just like focusing more on Isabel's question about like the difference between artificial intelligence and machine learning. Basically, artificial intelligence is like a more broader concept where like basically we are trying to make the program smarter, you know, and machine learning is a little bit like kind of a subfield where basically okay, your program is also smarter, but you are trying to make the machine understand by itself by giving data to it. So we have these concepts of what is artificial intelligence, machine learning, even what's the difference of machine learning and statistics and Bayesian statistics. And as Mia said, it's like a really great discussion, but basically we could say artificial intelligence is broader, machine learning is a part of it. And yeah, each one have its different specifics.
Thank you, Lucas. So, thank you, everyone. I know this was a bit short for discussion, but it was very interesting to hear you. Uh, Thank you for all the ones who presented your work. They were in different fields. You brought up different questions in that we could discuss about. And it was very interesting to have your point of view in this challenging field. Maybe this is just a beginning to start thinking and planning better our projects in artificial intelligence. So now the session is over, and I want to say just follow BSB tomorrow again. We have roundtables about in the afternoon about other interesting topics. And if you are also interested in this bioinformatics and artificial intelligence, try to join us next year in Mexico in an in-person conference and meeting. Okay. Thank you very much to everyone.
Thank you.
Thank you very much to everyone who attended. Bye-bye.
Bye.